At 1.6, cell division understandings. I'm going to let you read these, but I do use some abbreviations here. Capital M stands for mitosis. Capital C stands for chromosomes. So mitosis. Before mitosis, which is called interphase. Interphase happens before mitosis. All DNA needs to be replicated during interphase. So each chromosome is converted from a single DNA molecule into two identical DNA molecules called sister chromatids. So each chromosome after replication consists of two sister chromatids. They're genetically identical. Mitosis is involved whenever cells get genetically identical nuclei, whenever cells with genetically identical nuclei are needed in eukaryotes. This happens in embryonic development, growth, tissue repair, and asexual reproduction. I believe that might even be on the test. The events of mitosis are divided into PMAT, prophase, metaphase, anaphase, telophase. So we have interphase, then we have mitosis, which is PMAT. Interphase, again, this is before mitosis. The numbers of mitochondria in the cytoplasm increase by division, and chloroplasts in plant and algae. Interphase is in three parts, G1, S, G2. In G1, the organelles are replicated. S stands for synthesis of chromosomes, also known as DNA replication, and then G2. If cells don't divide, they stay in G1, and in fact, cells usually are not dividing. Our cells don't always want to divide. Often, they just stay in interphase. Okay, PMAT. In prophase, chromosomes become shorter and fatter by coiling. The nucleus breaks down. Microtubules form from microtubule organization centers that links the poles or the sides of the cell. It ends with the breakdown of the nuclear membrane. So the nuclear membrane breaks apart in order for these microtubules to attach to the chromosomes. Metaphase, M stands for middle. The microtubules attach to the centromeres of each chromosome. The centromere is the center of the chromosome. So one microtubule from each pole of the cell attaches to the centromeres in order to eventually break them apart. And they line up in the middle of the cell. A stands for away. So each centromere divides and their sister chromatids are pulled away to opposite poles of the cell. And telophase, T stands for two. Two nuclear membranes form around each group of chromosomes. All right, cyclins are these proteins that control the cell cycle. Cyclins bind enzymes called cyclet-dependent kinases. Kinases are enzymes that add phosphate groups or phosphorylate to other proteins, activating them to carry out specific tasks in the phase of the cell cycle. All right, tumor formation and cancer. Carcinogens are chemicals and agents that cause cancer. Mutagens are agents that cause gene mutations, which can cause cancer. Oncogenes are genes that can cause cancer if they mutate. They are normally involved in the control of the cell cycle. So if they mutate, the cell cycle may not be controlled, and then you have uncontrolled cell division, which is cancer. And metastasis is when cells from a primary tumor move and set up secondary tumors in the body. If that happens, it is very, very bad news. Guys, that is the end of the unit. We have a test on Friday. Good luck.